recognition of molecular drivers or molecular biomarkers or genes that are driving the progression of cancer, also comes the realization that there are many genes are involved, not only in different cancers, but they're also the same genes that are involved in multiple cancers. So if you look at these graphs, if you look at KRAS, for example, it's one of the driver mutations, for example, in multiple cell lung cancer and pancreatic cancer. And for example, if you look at, BRAF as another example, that's really important for colorectal cancer as well as melanoma. Not only are therapeutics being developed for these driver genes, so for example, defactinib for EGFR mutated non-small cell lung cancer, and this has been shown very clearly in a lot of cases, and this is just one example on the left, about the overall survival when patients are treated with defactinib instead of placebo if they are EGFR mutant. And the great thing about this is that the field has progressed on. A lot of new biomarker-driven drugs or biomarker-centric drugs, as you can see here, that have been given approval. And also, if you look near into the future, but with the different molecules and different genes that need to be looked at, the next generation sequencing technologies, and of course hotspot panels, but initially where we identify or parts of the genome or parts of genes were identified as to be important for certain cancers and to be assessed for certain cancers, and of course that expanded to genes, groups of genes and full genes, and so on and so forth. And now that has been expanded to a concept known as CGP, or comprehensive genomic profiling. We have seen adoption of tumor-specific hotspot NGS panel, and that sort of has morphed into a small panel, so to speak. I think in this day and age, a small panel is anywhere between 50 gene mark. That's kind of the definition of small panels these days. And again, these definitions keep moving, right? Because if you recall, say 10 years ago, a small panel is like 5-7 gene panel. And I think what's interesting and important about genomic signatures in cancer is it's rather complex to detect. It's not just looking at a single gene or groups of genes, but in essence, some of these require you to look at either parts of the genome or huge sections of the genome or sample across the genome. So if you look at the microsatellite instability, what you're trying to identify actually is the cells or assay if the cancer type or the cancer of interest of the patient has any mismatch repair deficiency. Now, if you look at the panel on the left, the right hand side, if there's a mismatch repair deficiency, this manifests as a MSIH or microsatellite unstable phenotype. So the question comes back to, are we able to identify all of these mutations or possible classes of mutations with one single assay? And the answer is yes. The concept called comprehensive genomic profiling is able to identify not just different nucleic acid-based mutations, such as DNA, RNA, as you can see on the screen here. So if you look at DNA side of things, you're able to identify single gene mutations such as SNPs, indels, rearrangements, copy number alteration. You should also be able to identify multi-gene biomarkers or genomic signatures, like what we just discussed, TMB, MSI, and HRD. On the RNA side, there's also the possibility of identifying known and novel fusions. We talked a bit about end-track fusions, as well as splice variants, and in some cases, expression balances as well. So the advantages of CGP is that it will optimize the yield of your actionable findings. It's one single assay using limited tissue, right? So as opposed to iterative testing, whereby you go single gene testing and you use up your very precious FFP tissue. It's also able to detect rare or infrequent variants, including fusion. And of course, there's broad genomic coverage for these calling of these genomic signatures.
I will first take the opportunity and to thank my lovely team. While it is a due-date conference, but it practically started to be planned 4 months back. Choice of the platform, going through the process of different items that should be covered in this conference. Attendees, we had 400 plus registrations, which was a decent number to start. Partnering with US India Business Council. So I think that the whole journey was really very satisfying. And this journey definitely was for a very noble cause. On a discussion of cancer, people do such a large charity and donations in this area. At least from our side, a good holistic, elaborative, comprehensive and interactive discussion from different research scientists across the world is our contribution for this particular cure of this disease. So with this as our concluding remark, with all those lovely comments and excellent effort, I'd like to personally thank Hitesh, our dear Kalol, and the rest of the team work towards it. And with that remark, I declare the conference now closed. Thank you so much, my dear friends.